Okay, today we're going to look a little bit at the DNA molecules and how they replicate. So the genetic code is the way in which the cell stores the program that they seem to pass from one generation of an organism to the next. And the genetic code, it contains the information for the cell to make proteins. And it tells the amino acids, tells the ribosome how to line up those amino acids while making the proteins. And it's the order of those amino acids that are going to determine the shape of the protein. And it's the shape of the protein that's going to determine the function of the protein. And it's the proteins being made in a cell that's going to determine that cell's functions. So that's all held in the genetic code of the DNA molecule. So the DNA molecule consists of a sugar called deoxyribose, phosphate group, and bases. When we looked at macromolecules, we looked at a nucleotide, and we said it had that. We had a 5-carbon sugar drawn, and then over here we had, sometimes we represented it as just a block. Sometimes we drew it out. We had a base over here, and that base can change. It may be adenine, it may be guanine, it may be cytosine, or it may be thymine. And then we had our little phosphate group over here. We said, well, those are going to be the building blocks of DNA. So here we are. We're building DNA with those nucleotides. And those sugars and phosphates are going to line up down each side of the DNA. DNA looks like a twisted ladder, so each side is going to have sugar, phosphate, sugar, phosphate, sugar, phosphate alternating. And then our rungs between our two rails, our rungs are going to be where those bases come together. Our adenines are going to combine with our thymines. Our cytosines are going to combine with our guanines. So A to T and G to C. So DNA is made up of repeating units called nucleotides, which consist of one sugar, one phosphate, and one base. And in DNA, that is deoxyribose for the sugar. Our bases are adenine, represented by A, guanine, G, cytosine, C, and thymine, T. So thymine and cytosine belong to a group of compounds called pyrimidines, and adenine and guanine belong to a group called purines. So purines and pyrimidines, thymine and cytosine. Thymine is going to go with adenine, and guanine and cytosine are going to go together. You always have a pyrimidine linking up with a purine. So thymine and adenine, cytosine and guanine. And this is what they look like. And notice that thymine and cytosine, they both have a 6 hexagon shaped sugar there. And then when we get to our purines, they have 2 rings. They have a hexagon and a pentagon. So purines, and it's kind of the opposite. Pyrimidines have a bigger name, but the smaller molecule. Purines have a smaller name, bigger molecule. So DNA is composed of 2 long chains of nucleotides arranged in a spiral called a double helix. Remember double helix. And again, it's like the rails of the ladder and the rungs of the ladder, and then it's just twisted. Somebody took a ladder and just twisted it. So it's a double helix. The sides of the double helix are made up of alternating molecules of sugar and phosphates. Sugar and phosphate are held together by covalent bonds. Now, it's important. The sugars and phosphates down those rails are held together by covalent bonds. Covalent bonds are pretty strong. The rungs, or the steps of the ladder, are composed of the bases. That's where your A is going to stick to your T, and your G's are going to stick to your C's. Adenine always bonds with thymine, and cytosine always bonds with guanine. Those bases are held together by hydrogen bonds, and that's going to be really important too. Hydrogen bonds are much weaker bonds. And as we see in a few minutes, we're going to have to open up those bonds. So it's a good thing that those are hydrogen bonds and easy to open. That allows helicase, that we're going to talk about in a few minutes, to come in and open up the bonds between those spaces so that a complementary pair can be laid down and we can copy that DNA. The 4 bases are said to be complementary because they go together. Complementary means that the bases on one strand determine the bases on the other strand. They complete the pair. They're complementary. And remember, the pair is AT and GC. So one strand of DNA is AT. This is our original strand. This will be our complementary. A is going to go with T. T is going to go with A. A is going to go with T. G with C. C with G. T with A. T with A with T, and G with C. So if this is our original strand, this would be our complementary strand. A always with T and G always with C. And I remember because G and C look an awful lot alike. G's just got this little extra thing here, so G and C look alike, so they always pair up together. Always. So as you can see from the model, the structure is basically a long, twisty, rope lettery type thingy. And there it is. First described by Watson and Crick. The structure of the DNA was discovered in 1953 by James Watson and Francis Crick, and they couldn't directly look at it, but they used methods of looking at shadows and bouncing things off of things to determine the shape of the DNA molecule. James Watson and Francis Crick first described DNA in 1953. So the functions of DNA determines which proteins will be produced by the cell. The type of proteins determines the activity and physical features of that cell. It's going to determine if that's a skin cell, if that's a lung cell, if that's a neuron. So it's going to be determined by the proteins that are being made in that cell. The sequence of amino acids in the protein chain determines what kind of protein it is. And we said amino acids were the building blocks of protein, and there's 20 amino acids that we know about, and all proteins are made from those same 20 amino acids. They're lined up and held together by peptide bonds, and it's the order that's going to determine the shape and the activity of that protein. So a series of 3 nucleotides, or 3 bases on the DNA molecule, is called a triplet. So I said a DNA triplet. The triplet codes for an amino acid. So for example, ATT would code for a specific amino acid, and then a little further down we might have a GCG would be your next 3 bases, and that would code for amino acid. Then the next 3 bases might be a T and an A and a G, and those would code for amino acid. And then the ribosome, that code is going to be read through a RNA molecule that we'll talk a lot more about when we get to protein synthesis, but those are going to code for the amino acids, and the ribosome is going to line up according to that original code in the DNA. So therefore the sequence of bases in the DNA make up the code for specific amino acids in the protein chains. And when cells divide to make new cells, each new cell must contain a complete set of that genetic instructions, an exact copy of the DNA molecule. The DNA code is copied by the process of replication. DNA replication. So how does replication work? DNA replication is carried out by a series of enzymes called DNA helicases and DNA polymerase. These enzymes, DNA helicase, is going to unzip the 2 strands of the DNA of the double helix. Remember I said a minute ago that the bases were held together in the middle by hydrogen bonds, and that they were not very strong bonds? Well, this is why that's important, that DNA helicase is going to have to come in and unzip between those bases. The DNA polymerase will then, after the helicase is unzipped, the polymerase is going to come in and insert those bases, those complementary bases. If there's an A, it's going to insert a T. If there's a G, it's going to insert a C. If there's a C, it's going to insert a G. If there's a T, it's going to insert an A. So A's always go with T's and G's always go with C's. And that polymerase is going to come in and lay down that complementary strand. And then it's going to proofread it. DNA polymerase proofreads the bases to ensure that they are pair correct. Occasionally mistakes happen, but the job of that polymerase is to put in those bases in the correct order and then proofread and make sure that they are correct. And this is what that would look like. Helicase is going to come in, and it was like this. It was the ladder together. Here, helicase is already unzipped. The helicase is going right here in front of the polymerase. Helicase is going up through here and unzipping. Polymerase is coming along behind it and laying down those complementary bases. There's a G here, so it put in a C. There's an A here, it put in a T. There's an A here, it put in another T. Here's a C, so it's going to put a G next. Here's a T, so it'll put an A next. And it's doing it up one side and down the other. It's pairing those up. Over here we have an A, so the next one there will be a T. We have a G, the next one's going to be a C. So the polymerase is putting in. Polymerase is going to put in and proofread. P's go together. Polymerase will put in and proofread. Helicase is unzipping it like a suitcase is unzipped. Helicase, suitcase, unzips. Polymerase puts in and proofreads. Polymerase puts in and proofreads. And here's another picture. This was our ladder. These are our rungs. That's the sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And those are held together by covalent bonds, stronger covalent bonds. And then here are our bases and our rungs, our A's to T's, our C's to G's, T to A to T, G to C. And these are held together by those weak hydrogen bonds, which is really important because then the DNA helicase can come in and unzip it. And then our polymerase is going to come in and put in these bases. G's to C's, A's to T's, A's to T's, and then you're going to end up with two pairs. See how this was AT, now we've got an AT here and an AT here. We made a copy of what it was. That polymerase is putting in and proofreading those bases. When the DNA in the cell replicates, the cell has two exact copies of the DNA molecule. The cell then divides with each cell getting one copy of the DNA molecule. So then we'll end up with two identical cells with identical DNA. So why is replication so important? DNA replication is necessary to life because DNA contains the genetic information needed to form and control physical makeup and chemical processes of an organism. And I determine the function of each of those cells. Okay. So sample problem. If this is our original DNA strand, what would our complementary strand look like? And of course, it won't let me write on it, but we would have A to T, G to C, C to G, T to A, T to A to T, G to C, C to G, and G to C. And that would be our complementary strand. Quiz time. The primary function of DNA is to provide energy for the cell, transmit messages between cells, translate the coded RNA, or store information for building proteins. Now, the correct answer is going to be D, store information for building proteins. The primary function of DNA is to store information for building proteins, which is then going to determine what that cell does. The structure of DNA was discovered in 1953 by Watson and Crick. Watson and Crick discovered the structure of DNA. So what type of bonds hold the nitrogenous bases together in the DNA molecule? The bases in the middle, those nitrogenous bases, are held together by hydrogen bonds, weak hydrogen bonds that allow the helicase to unzip. Cytosine and thymine are okay, we know they have to be either purines or pyrimidines. So, do you remember which one? Cytosine and thymine are the pyrimidines. Cytosine and thymine are pyrimidines. And that means that purines, that would be the adenine and the guanine would be the purines. Cytosine and thymine are pyrimidines. And they have Y's in them. Cytosine has a Y, thymine has a Y, pyrimidine has a Y. That will help you remember. The Y's go together. Cytosine has a Y, thymine has a Y, pyrimidines have a Y. So cytosine and thymine are pyrimidines. So what is the shape of the DNA molecule? That will be double helix. The shape of the DNA molecule is the double helix. Twisted ladder, properly named double helix. And that's that. Have a good day.